Hey y'all, this is Mr. Johnson. I am going to run you through the chapter 8 and 9 notes really quickly. Hope you guys are doing well and washing your hands. Chapter 8 and 9 is on from DNA to proteins. So we call that the central dogma. A central dogma is anything that is a central idea or thought from anything. So in science, our central dogma is how we get from DNA to make proteins. What proteins are what we're made of. So we start with kind of a history lesson first. We've got a couple scientists. First, we have Frederick Douglass. He found that material, some sort of material, is being exchanged between organisms. So Frederick Douglass discovered that information was being exchanged, and we call this transformation. So transformation is when DNA is exchanged between organisms. So we have two different types of bacteria. The S bacteria, when injected into a mouse, kills it. The R bacteria, when injected into a mouse, doesn't kill it. When we heat kill the S bacteria, which essentially means we just heated it up, the mouse lives. So after we've heat killed the bacteria, it still lives. But when we heat kill S bacteria and insert live R bacteria and insert both of those things into a dead mouse, or into a mouse, that mouse dies. So what's going on here is that heat-killed S bacteria, the membrane on the outside is breaking down. The R bacteria is taking up the bad parts of the S bacteria, the parts that kill the mouse, and transformations occurring, right? Okay, it's exchanging DNA. Okay, so who actually found that DNA? I'm going to skip this. Was genetic material. We have Hershey and Chase. So Hershey and Chase confirmed that DNA was the genetic material. And you might think, well, duh, DNA is genetic material. Well, we didn't know that. We thought it could be RNA. We thought it could be DNA, right? And so on and so forth. And here we've talked about bacteriophages before. We drew something very similar to this when we talked about viruses. Bacteriophages are just specific viruses that infect bacteria. Okay. And maybe I shouldn't mention the word viruses, PTSD for some of us. So we have a couple scientists that came up with the structure of DNA. So our structure of DNA also was something that had to be discovered. We didn't always know it was this. So Watson and Crick are two main people that came up with the structure of DNA, but there's another person that didn't get a lot of credit. Oh no, I need to get rid of this. How do I get rid of that? Bear with me, all this PowerPoint is rough. Rosalind Franklin is someone that doesn't get a lot of credit, but she used a process called X-ray crystallography to accurately depict the image of DNA. And I think I deleted the image, but it's a cross section of DNA, and it shows the 3D structure of it. So what process did Rosalind Franklin use? She used X-ray crystallography. Okay. Another scientist we have is Erwin Chargaff. Erwin Chargaff came up with our complementary base pairing rules. So what does it mean to be complementary? It means that things go together. Okay, so peanut butter complements, right? Oh, complementary. What do we say it complements? Probably jelly, right? Peanut butter and jelly, they complement each other. Singing and dancing are complementary. I don't know, y'all. I'm out of brain power right now. Okay, all these things are complementary. So Erwin Chargaff found that there are certain pairs of bases that complement each other in DNA. So we need to talk about those four bases. So we have four nucleotides or four different nitrogenous bases. Okay, this is what a nucleotide looks like. It contains the nitrogen-containing base or the nitrogenous base, a phosphate group, and a sugar. We call that sugar deoxyribose, which is where we get the name DNA, deoxyribose nucleic acid from, okay? Our first one is adenine, that's nucleotide. So under nucleotide, you can write adenine. We just say A, okay? Adenine is a double ring. Any double ring is called a purine. Those two things are the two descriptions you can write. And adenine always pairs with thymine. Thymine we have as T. So thymine's the next nucleotide. You can write under nucleotide as thymine. Thymine is different from adenine. Well, adenine is a double ring, thymine is a single ring, and we call it a pyrimidine. Okay. And thymine always pairs with adenine. They complement each other. Okay, you may want to pause me because I'm going to roll. We've got guanine. Guanine is just like adenine, so guanine is a double ring, and we call it a purine. It looks very similar. Guanine is complemented by cytosine. Cytosine is a single ring, and it's just like thymine. Since it's a single ring, we call it a pyrimidine. It looks like that. Okay. So what joins these nucleotides together? Well, we have hydrogen bonds, which are like one of the most important bonds in life in general. So between adenine and thymine, A and T, we have 2 hydrogen bonds. Between guanine and cytosine, we have 3 hydrogen bonds. And we see how these things are always complementing each other. Where we have a T, the opposite side has an A. Where we have a G, the opposite side has a C. We see that going all the way down here. If that didn't occur, we call that a what? We call that a mutation, right? Okay. Now, if we know how much of one of these nucleotides we have, we can figure out the rest of it, right? Okay, so that's where Erwin Chargaff's base pairing rule comes in. He said that adenine always pairs with thymine and cytosine always pairs with guanine. So if we know something, right this, a scientist takes a sample of DNA and finds that it contains 20 nucleotides of adenine, how much thymine will it contain? Well, it's going to contain 20, right? And why? Because adenine complements thymine. If there are 20 adenine, they will be complemented by 20 thymine, okay? Another example, we have a scientist takes a sample of DNA and finds that it contains 50 nucleotides of guanine. How much cytosine will it contain? Well, if it has 50 guanine, it has to contain 50 cytosine. If it didn't, it's a mutation, right? Okay. We can go a little deeper than that. If we start talking about percentages, we can figure out all the percentages of these nucleotides, okay? These percentages will add up to 100%, and the complementary bases will be equal, okay? So it says a scientist takes a sample of DNA and finds that it contains 15% thymine. How much adenine will it contain? Well, if it contains 15% thymine, it's going to contain 15% adenine. And let me stop there and ask, can you figure out how much cytosine and guanine will be? Okay, if it has to add up to 100%, we know that 100 minus 15 is 85. 85 minus 15 is 70. So the remaining percentage down here should add up to 70, and it should be equal. So 70 divided by 2 is going to be 35. 35% right here, okay? Let's do another example. It says a scientist takes a sample of DNA and finds that it contains 45% thymine. How much cytosine will be in the sample? Okay, we know that thymine is going to be 45%. We know that adenine is going to be 45%. So what percentage is left over? Well, this up here together is 90%. That means we only have 10% left over to split between cytosine and guanine. 10 divided by 2 is going to be 5%, right? Okay, so cytosine is going to be 5%. Guanine is going to be 5%. Okay, and that's how you do some base pairing with Erwin Chargaff's rule. Okay, and here's another example. If we have 10% cytosine, we're going to have 10% guanine, 40% thymine, and 40% adenine. That should make sense, right? Now we get into the processes of replication and how mutation comes about. So the process of DNA being copied is called replication. The goal of replication, oh no, again, y'all, again, this is bad. The goal of replication is right down here, hopefully. The goal of replication is to create 2 identical strands of DNA. And why would we need to create 2 identical strands of DNA? Well, it's because we're dividing in mitosis, typically, right? That's why we're doing replication, okay? The process of replication includes 3 enzymes. We know it's an enzyme because they end in ACE, DNA helicase. We want to imagine that it unwraps the helix. The double helix structure is unwrapped in DNA helicase. Next, we have DNA polymerase. Polymerase brings in new nucleotides. So if polymerase reads adenine, it's going to bring in the one that complements adenine, which is thymine, T. If it reads a C, it's going to bring in a G. If it reads a T, it'll bring in an A, and so on and so forth. After DNA polymerase creates these 2 new strands, those 2 new strands have to be sealed up with a glue. We call that DNA ligase. That G reminds us of glue. It zips up the DNA and glues it back together, okay? We call this process semi-conservative, and semi-conservative means that one half of the strand is conserved and another half of the strand is new, okay? So we never have one entire strand that's new and one entire strand that's old. It's half and half, okay? And replication happens quickly and accurately, and mistakes that are made are typically fixed. And if they're not fixed, what do we call them? We call those things mutations. Those are changes in DNA, and we have 3 different types that I'll run through. We have substitutions. Just like if I'm absent one day, you'll have a substitute, right? It's when one one nucleotide is changed with another nucleotide. So we see our strand of DNA here, the C gets changed to an A. That's a substitution. Does it affect a lot of things? It might, but it doesn't typically. These things kind of happen pretty often. Next, we have insertions. Okay, insertions are not good. Insertions are when a nucleotide is inserted or places itself between other nucleotides. That causes a frame shift, and these are big deals. Deletions are exactly what they sound like. A nucleotide is deleted. Okay, and there is not just these three types of mutations. There's other types of mutations, but we are not going to be able to talk about those things for obvious reasons. Okay, mutations we can talk about can cause a lot of things. It can cause a gene to work incorrectly, right? It can cause an incorrect protein to be formed, inability of offspring, and sometimes there are beneficial things like sickle cell anemia and Marfan syndrome, and I don't know if we've talked about those things at all. But perhaps I'll have time to talk about those things in another video. Okay, I'm going to take you through the central dogma really quickly. The central dogma is this right here. It's where we go from DNA to RNA to proteins. We cannot go straight from DNA to proteins. We need an intermediate, RNA, because DNA cannot leave the nucleus. So RNA acts as a messenger to get out of the nucleus and create proteins. So all these processes here are essentially everything in the central dogma and the rest of your notes, essentially. Okay, so RNA is a temporary copy of DNA. It allows it to leave the nucleus, and it's a little different. Oh man, it's a little different. Okay, let me try that again. It's a little different from DNA in that it has, instead of a T, it has a U. This is uracil, okay? It's also single-stranded, so one of the main differences in RNA structure and DNA structure is that it's single-stranded. It's not double-stranded, which helps it to get out of the nucleus a little easier through those pores, okay? And I wish I was there to show you the pores in the nucleus, because I don't know if I got a chance to show you that in chapter 3. So the first process of the central dogma is transcription. Transcription is taking DNA and making RNA, okay? So RNA polymerase helps to bind the new nucleotides to make RNA, okay? It's just like DNA polymerase, it brings in nucleotides to make RNA, okay? There are 3 parts. First, one gene is unwound, not the whole DNA molecule, just one gene. RNA polymerase creates a new RNA, and the DNA strand gets wound back up, and the RNA gets to just pop out and leave the nucleus, okay? We have 3 types of RNA, and this is something I would spend a lot more time in class, but I do not have time for it here. Here are the 3 types if you want to write those things down, okay? So the goal of replication is to create 2 identical strands of DNA. The goal of transcription is to produce RNA. Once we produced RNA, that's not the last thing that we need to do. We need to do translation, and translation is converting RNA to proteins, okay? When we convert RNA to proteins, the ribosome is reading, and I don't know if this is correct. RNA is reading nucleotides in groups of 3 called codons. Okay, so this should, your notes should say, number 29, nucleotides in groups of 3 are called codons, okay? A start codon signals the beginning of a chain, and that is AUG. The nucleotides A, adenine, U, uracil, G, guanine, signify the start of translation, okay? Translation occurs, where does it occur? Translation occurs at the ribosomes, and tRNA carries the amino acids to the ribosome to form a protein, okay? And this is the central dogma. There's a couple things that we can do with DNA. One is gel electrophoresis. We can separate DNA fragments and read them and see how similar or different they are. Polymerase chain reactions, not a process I have time to explain right now. I'm running out of time as I'm speaking. And all people have different DNA fingerprints. If you've seen CSI, then you know that DNA fingerprinting is important for that. Blah, blah, blah. I'm running out of time. I literally have 10 seconds right now, y'all. And the last thing, oh god, genetic engineering, I'm not going to get to. I'll post another video next week at some point. I love you guys. Talk to you soon.